I called on you for your questions about plant science and genetics, and you answered the call with your questions. So I'm going to answer those questions. So yeah, basically, a lot of questions and answers in this episode. How many times can I say the word question in the space of a minute? Let's find out. Anyway, from ant plants to how tulips caused the first speculative financial bubble, we've got some good stuff, so let's dive into it. Welcome to Modified. I'm your host, Dr. Orlando Delange, ex-plant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs and hopefully just enough of the history and the social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 5.5, Your Questions Answered. I had wanted to do something like 10 questions, but I realized that it would probably be more satisfying to be able to do a deep dive answer for each question, and so I picked my 3 favorites. But I am hoping to do more episodes in this style in future if you would like that, so just let me know. And just to mentally prepare you, you'll notice I don't sound so fully scripted in this episode, and that is because I'm not. I did some research, I had some idea of the things to talk about, but I'm speaking more off the cuff as I go through, so a little bit of a different style. Hopefully that doesn't throw you off. So, question 1. What is the weirdest plant defense mechanism scientists have discovered? I want to thank @LondonMutt on Instagram for this question, because it allows me to just ramble on about cool plant stuff, which I'm always happy to do. So I think there are a few mechanisms that are reasonably well known, but that doesn't mean they aren't really weird and worth talking about. So I wanted to pick a couple that I think you probably have heard of, and then one that you probably haven't. So the first one that you probably heard of is Mimosa pudica, aka the shy plant. And it has a pretty special defense mechanism, which you might know about, you might not know. It's a plant that grows kind of as a weed. It's quite a scraggly little species, and it's native to Central America, but these days it's found all over in kind of tropical and subtropical areas. It's a big genus that mimosa comes from, and it's part of an even bigger family, which is the legumes, which includes all beans and peas. You may know that the legumes are unique in the plant kingdom for forming a special symbiosis with bacteria in the Rhizobium genus that are able to pull nitrogen from the air in its inert form, what we call molecular nitrogen, which is about 80% of the air around us. But unfortunately, most plants can't use nitrogen in that form. They need nitrogen in an organic form, something like nitrate or ammonium, where it's basically the chemical bonds have been modified in a way that makes it kind of accessible to normal biochemical processes. And nitrogen is often really limiting for plants, so it's incredibly useful for legumes that they house these little bacteria, these rhizobia, that have some kind of biochemical wizardry where they can actually pull nitrogen from the air and what we call it fixing, which is like convert it into an organic form that can be used by the plant. And in return, the plant provides a physical housing within the roots, like you can pull up the roots of a legume, and you'll find these little nodules, these little round balls that are a little bit pink in color because of an enzyme in the bacteria. And that's where they physically house the bacteria. So those bacteria have like a kind of a good environment to grow in, and they get a bunch of sugar from the plant. And in return, the plant gets this leg up over all the other plants around it in the form of easy access to nitrogen. So that's just a little thing about legumes. That's not the specific thing to do with Mimosa pudica. So the Mimosa pudica defense mechanism we're going to talk about is not defending itself from the rhizobia, but defending itself from herbivores. And this is a defense mechanism that has been well known for, I'm sure, thousands of years, since as long as humans and this plant have interacted. But actually, despite thousands of years of kind of human knowledge of this kind of the existence of this mechanism, the details of how it works on the molecular level are really, there's been recent breakthroughs, and there's still quite a lot of open questions about it. And so if you don't know a lot about plants, you probably do at least know that they are not renowned for their fast movements. I actually think it's useful to think about plants as organisms that do move, but they move via growing. By growing in particular directions, that's how they kind of reach out and access particular, maybe nutrient pockets, or better sunlight or water. But it takes time. So depending on the plant and the plant organ, growth might take place over several hours in the fastest cases, or several decades in the slowest. Normally, it's something more in between over the space of days and weeks of active growth. You've probably seen this, like, most of the time plants, they're not really look like they're doing very much, but when growth is active, particularly notable in temperate regions in spring when new leaves are coming, they're kind of budding and expanding, you can see very dramatic growth over the space of a few days normally. But so that's, in general, plant movement pretty slow, but there are a few exceptions when it comes to this, and one of the most famous is the Venus flytrap. And then another is the one we're talking about, the rapid folding of the leaves of the Mimosa pudica plant. Mimosa pudica has pinnate leaves, like many legumes, and so you could Google that, but think of it like feathers. So they're kind of long leaves with lots of little leaflets all along the side, instead of like the individual fibers of the feather. And all it takes is a single touch. So you can go up to one of these plants, touch it in one of its leaves, and you'll see all the leaflets along that plant, along that leaf, folding up. And then that same process spreading within a few seconds to all the neighboring leaves, and all the leaves along that branch will fold up, meaning instead of kind of being held out horizontally, they will droop vertically down. And this is very dramatic, very obvious and visible, and happens within the space of a few seconds. And that is pretty rare and unusual in the plant world. How this happens has been known for a while in a kind of physiological way, kind of like in the sense that it's known that there's a little structure at the base of each leaflet called a pulvinus that looks like a slight bulge at the base of the stalk of each leaflet and each leaf. And you can see this with your eyes if you look at the plant. And inside that pulvinus are hundreds of cells that are very densely packed together. And under normal conditions, they're pumped full of water, which holds them very rigid and holds the leaflet horizontal. However, with the right signal, proteins called aquaporins inside the membranes of those cells are activated, and that allows water to flow out of the cells quite rapidly. And as they lose water, they lose rigidity, and that pulvinus suddenly loses its tension, and the leaflet drops. And then it's a reversible reaction. So you don't need kind of irreversible growth. It's something you can basically pump water in or out of those pulvinus cells, and that's what allows the leaflet to droop or kind of return to its rigid horizontal position. It's also known, or at least it was kind of assumed, that it must be that there are some receptors, so specialized cells within the leaflets called mechanoreceptor cells that trigger an electric action potential that can rapidly travel to all the pulvini so that's plural of pulvinus, in the vicinity of the touch, telling them to kind of close up shop. And this is just because that's similar to how a Venus flytrap works. We know that plants have mechanoreceptors that normally don't respond very quickly, but they are definitely sensitive to touch, and they have these specialized cells to do that. And it's known that plants can exhibit action potentials, which action potentials are the same way nerves work in animals. Essentially, there are proteins in the membrane of basically every cell, whether animal, plant, or other, that regulate the flow of electrically charged ions. And for any given cell, there's a resting balance of ions that is constantly actively maintained. But under certain conditions, the regulator proteins change their state and allow ions to flow in the opposite direction, either by removing barriers that have held ions back or by using energy to pump ions in a particular direction. And that changes the electric charge of the cell, which itself is a powerful signal that can be detected by proteins in neighboring cell regions. And that tells them, okay, you should open up the membrane channels, the transporters, and kind of perpetuate this signal. And that in animals, we have these long specialized cells called nerve cells, where that action potential signal can pass along the whole length of the cell. And we have individual nerve cells that can go up like a whole leg. They can be very, very long. And they have a bunch of specializations kind of around nerve cells that they allow the action potential to propagate very, very rapidly. Plants don't have cells like nerves that are specially adapted to propagate action potentials. It's more every cell can propagate an action potential to some extent, but they move slower than in our cells. So in the space of seconds instead of milliseconds. But one of the things that plant cells do have, which facilitates, instead of having, one giant specialized nerve cell that the action potential can propagate along the length of, all the cells in an average plant organ are actually interconnected with each other via tiny cell-to-cell linkages. So where there's actual direct connections where the membranes have fused and there's no barrier in between the cells in very small little areas dotted around the connection surface between the cells. We call those plasmodesmata. Anyway, these little connections, they have a bunch of different functions in plants, but one of the things that they allow is for the fairly rapid exchange of ions between cells, and that exchange, that kind of facilitates the movement of the action potential. So this was already known, or we're not sure. At some point early 20th century, I think people found out that action potentials existed in plants as well as animals. Okay, so the piece of the puzzle that connects back to the listener question that has been harder to pin down till now is, why does Mimosa pudica fold its leaves? And this is something that there has been some kind of recent research published on, but there just was a complete dearth of research, scientific research published in this area till now. And I think it's kind of like worth reflecting on why. And I think there's a few reasons. One is that plant science research is very expensive. Really, any life science research these days is very expensive. It requires a lot of people. It requires specialist equipment that all costs money. So research tends to be focused on things with real-world applications that a lot of people care a lot about. And for plants, that means overwhelmingly agriculture. No one eats Mimosa pudica, to my knowledge, so there's just not that much incentive for anybody to bother investing a bunch of money into research into it. And that means for the vast, vast majority of plants, we know very little about them other than maybe the basics of their morphology, features like descriptions of their flowers, enough information that you need to kind of classify them and say, okay, we kind of know what type of plant this is and how it fits into the plant family tree. But there'll be, for most plants, no one has ever done a kind of like detailed biochemical analysis on their different cell components and looked into their genetics and all sorts of different things that we have the technology to do, but it's like it takes time, it takes money. Mimosa pudica, we actually, have some research into, I think probably just because it is such a cool, fascinating plant that has this feature that people are just quite interested in. Not interested enough to want to pay a bunch of money to support a bunch of research teams, but enough to maybe pay a couple of research teams around the world. So anyway, there's a little bit of just general plant science background. But there has been some recent research that I wanted to summarize. So Takuma Hagahara and his colleagues from several different institutions in Japan, as well as the University of Wisconsin-Madison, published a data-packed and fairly easy to follow paper just a few years back in 2022, which I will, of course, link in the show notes. And one of the first things they did was document a detailed experimental investigation into why Mimosa pudica has this rapid leaf drop. And they, from what I can see, were the first folks to do so in a scientific paper. So to really say, why does the plant have this physiological process, you'd need to rewind evolution over millions of years and be able to chart all of the kind of conditions in the environment of the plant that it was responding to through natural selection, or the natural selection was responding to. We can't rewind evolution, so all we can do is kind of make some reasonable guesses by seeing, okay, well, making some assumptions about what do we think is the advantage for the plant, and then test those assumptions. And so that's what they did. They already assumed that the reason that the plant has this behavior, we could call it, is to defend it against herbivores. And so but they're still the first people to actually test that experimentally, to my knowledge. And so they checked this in a couple of different ways. And one of the ways they did this, and I should have some sort of GMO klaxon, is that they made a genetically modified Mimosa pudica plant, which is quite exciting. And it's also, not to keep digressing too much, but I think this also is a good chance to check in on the fact that most of the genetically modified plants that are produced around the world, not in terms of like total volume of plants, but in terms of like all the different species that have been genetically modified. There's very few genetically modified species or varieties that are for commercial application, but there are tons and tons and tons made all the time for research purposes. And I know this because it's, the sort of thing I used to do. Most molecular plant science labs these days, one of the tools among many they use is genetically modifying plants to kind of ask questions about them. So in this case, they use genetic modification, and they use a thing called CRISPR, which we'll learn about more at the end of this season, but basically allows you to kind of go in and break specific genes within the plant genome. And so what they did was knocked out, is like, the technical term that we use, knocked out the gene, broke the gene that is crucial for the function of the pulvinus. And what this means in practice is it basically stops the plants from being able to have this drooping effect. And they also stimulated the same thing using a different method, which is like a great piece of scientific technique, is like make sure you can ask the same question using two independent experimental methods, just in case there was something kind of like an unexpected side effect of your first experimental method, where it kind of gives you a result and you think it's because of X, but actually it's because of some hidden feature of your experimental setup. So it's good to like try to ask the same question multiple different ways. So they made this genetically modified version of the plant. They also just used a chemical that stops the pulvinus working. And in both cases, they looked at how long potential herbivores, in this case it was some caterpillars and some crickets, spent feeding on the leaves. And they just kind of recorded that and were able to show fairly clearly that plants that lack this ability to droop were basically had their leaves eaten more. It's pretty much as simple as that. And it seems like quite a simple experiment, but it's like you do have to kind of do it carefully and make sure you're really tracking it and have these controls in place. But for as far as I can see, they were the first to say, like, yes, okay, maybe this isn't exactly why it evolved, but we do know that at least this drooping mechanism, at least in a laboratory setting, does reduce herbivory on these plants. So anyway, that's something that I thought was very cool. Another kind of cool defense mechanism I wanted to mention, but I'll just touch on very briefly, so I can get to my favorite one, is ant plants, defense by ant. So that's right, there are in fact hundreds of species of plants, mostly in tropical and subtropical parts of the world, which have a mutualistic symbiosis with ants. Mutualistic symbiosis meaning they living very, very closely and intimately together, and each side benefits. Just like the rhizobial legume symbiosis we talked about, that's mutualistic. So is the ant-plant symbiosis. Depending on the species, the plant might provide a special structure that houses the ants. So this is not just ants kind of happen to be near to the plant. They live inside the plant most of the time. And in most cases, the plant also has a specialized organ that provides food. This can happen in various different ways, but it's normally like little nodules, little food packets that the plant produces for the ants. And in return, the ants fiercely defend the plant against any would-be predators or herbivores that get too close. And there's a fun word for this, myrmecophytes. Literally ant plants. But doesn't myrmecophyte sound cooler? So yeah, go Google myrmecophytes and look at some of the really cool kind of ant houses that these plants grow. But in the interest of time, I will move on to the next one, because otherwise this episode might be super long. I wanted to end this long answer to still our first question with my favorite weird plant defense mechanism, the chameleon vine. So chameleons, these crazy lizards that can change color to mimic their backgrounds and blend into their surroundings. Well, in 2014, a bombshell hit the botanical community. Two researchers from Chile, Ernesto Giannoli and Fernando Carrasco Ura, published a report of a tropical vine, Bucilla trifoliata, that, unique among all known plants, can do what chameleons can. Presumably, this property has been known to those living in and around the native forest of this vine for a very long time, but the native range is not that far, it was not known to that many people before, and there had been no scientific reports published on it that can help. One of the benefits of a scientific paper is it's just a kind of way to easily alert researchers around the world to something. And so in 2014, they published a paper. They're not just said, okay, the vine exists. The vine had already been described briefly before, but what they showed was that this vine has a chameleon kind of property. And rather than changing color, what this plant can do is that the leaves of this vine change shape and also their kind of texture and color, depending on the tree that they're growing on. And I'm going to link the paper again in the show notes so you can look it up. And it is actually quite astonishing. This is not like some super subtle thing where you have to kind of squint and you're like, okay, I guess those leaves are like maybe a little bit more round. No, this is quite dramatic. It's not a perfect camouflage, but it is pretty good. And like, it's so amazing that you could even have one Bacilla trifoliata vine that happens over its whole length to be growing on 3 different trees at different points along its kind of main branch. And it will have different shaped leaves at each of those points based on the tree that it is growing on. So they're able to show this isn't like there's just some kind of variation in the morphotypes of this plant. That's a fairly common thing, that if you kind of have a field of plants, within that field you might find different versions with different leaf shapes. No, it's literally one individual can have different leaf shapes, and that if you experimentally grow the vine in a lab, give it different kind of host trees to grow on, you can see that it does indeed adapt its leaf shape to the vine that you give it. Even artificial vines that don't exist in nature. Sorry, artificial host trees. And this kind of blew everyone's mind because there's just no precedent for this in the plant world. And so that was in 2014. And they did, a key thing I wanted to point out is that they also in the 2014 paper showed that this does indeed reduce herbivory. So the kind of very similar to the previous paper, they're basically able to show that this property does reduce the amount of time that insects spend. Basically, what it seems like is that the insects notice the leaves less. Just like with a chameleon, it can kind of like blend into the background more. But they were not able to show how this mechanism works. And I would say, like, there's a couple of updates that I'll give you, but it's still very much an open question. I do not think it's been definitively solved at all. And there was a couple of, like, main approaches or, like, theories about this. So in 2020, an American amateur researcher, which I think is really cool to shout that out because these days it's very hard to kind of participate in scientific research unless you work, unless you have a PhD and you're working within some kind of traditional institution, but it can be done. So there was an American amateur researcher teamed up with a German researcher at the University of Bonn to show that the vine will attempt to mimic a plastic vine that it's grown next to, and that they propose that this is due to plant vision, which is not as crazy as it sounds. No one is suggesting that plants can see like animals can, but plants can certainly sense and respond to light. And what they're hypothesizing is that these plants have specialized cells that are particularly sensitive to light and can detect basically roughly the shapes of the leaves that they're growing near. And then kind of there's a feedback mechanism to recreate that shape, which, that ability to kind of basically see shapes would be unprecedented. But they did not find these cells. They did not show that mechanism. They were more showing like, well, it seems logical because we can give it some shape it's never, encountered before in its evolutionary history, and it's still able to mimic that shape. So it must be able to somehow get information about the shape in real time. And also, it was a plastic leaf. It wasn't giving off any natural plant chemicals that might be, the other main theory is, okay, each of the different trees that happens to grow in the native range of this vine has a certain kind of chemical signature to it. So all the plant needs to do, the chameleon vine, is detect that chemical signature and kind of like switch between its different pre-programmed leaf shapes. But that does not seem to be the case. And so another paper came out in 2022. So like this is all very recent stuff, still very much an ongoing question, showing that there seems to be some overlaps in the leaf microbiome of Bacilla trifoliata and its host trees, suggesting that microbes growing on the leaves provide a signal to the vine to start mimicking. And like as I just pointed out, that doesn't seem to stack up with the fact that it can also mimic some random plastic leaf shape, because the plastic leaves will not have microbes growing on them, at least they shouldn't. And so it doesn't seem to make... And also this other group with their kind of hypothesis that something about the microbiome of the leaves, they haven't got a mechanism for that yet. Like, this is still a very open question. So if you're like, wow, chameleon vines, that sounds amazing, get involved. There's plenty of open questions there. Like I mentioned, you don't expect to get a bunch of government funding to study this, because there's really, no one eats this chameleon vine. With that said, sorry to get on my soapbox, but, this, I think, could end up being a classic example of where basic research into something that seems very niche and obscure could pay off. Because whatever it is, there seems to be some totally novel kind of physiological mechanism in this plant, and if you can work out what that is, who knows, maybe it will have some really interesting, very practical applications. But you don't know until you find out. Anyway, so that long rambling journey will wrap up question 1 for now. Okay, questions 2 and 3 will not be quite as long, I promise. Okay, the next question: are dahlia speckles like tulip distortions and caused by a virus? So I love this question from @FCK Iceberg Lettuce on Instagram, because it gives me a chance to talk about ornamentals, aka flowers, which have some really interesting botany and history. And if any of this sounds interesting to you, the stuff that I'm about to talk about, you can read a lot more fun flower stories, the intersection of history and botany, in Flower Confidential by Amy Stewart, a really enjoyable read. I recommend it. So, but let's dive into it. So the question has 2 parts. It's the dahlia speckles, and are they like tulip distortions, which are caused by a virus? So I'm going to start with talking about the tulips, because some of you may know about this, but some of you probably do not. There are a lot of things to talk about with tulips. They have a lot of kind of juicy history to them. You probably know tulips. They have these bowl-shaped, richly colored flowers, and their name, in fact, comes from, or the name in English, from the Turkish word for turban, because of the resemblance between the flowers and a kind of luxurious turban. And that's, particularly fitting because they were introduced into Europe from Central Asia via Turkey in the 16th century. By that point in the 16th century, the wild tulip flowers from Central Asia had been selectively bred, for a long time in Central Asia to produce beautiful ornamental flowers. And so, by the time they arrived in Western Europe in the 16th century, these were not, the wild flowers. This was already a domesticated ornamental, and it really blew people's socks off. People fell in love with these tulips, and in particular, the Dutch went crazy for tulips, reaching a peak known as tulip mania in the early 1600s. So this is about 400 years ago. And it was, within a century of tulips first arriving in Western Europe. Like, they were a pretty overnight sensation. Tulip mania kind of peaked and already, as we'll see, collapsed within the space of a century. I am not sure where the cause and effect lie, but ever since tulip mania, so around that kind of like early 1600s, the Netherlands has been a global center for floriculture, meaning the cultivation and trading of ornamental flowers, and they are still the global leader in that industry. I don't know quite what it is, whether tulips are really anything to do with it, or it's kind of like they're just another symptom. Dutch people and floriculture seem to just really go together. So tulip mania wasn't just a name for a craze, but is normally used to refer to a speculative bubble that happened kind of towards the end of this period of intense interest in tulips, as people started to trade tulip bulbs at higher and higher prices, and in a way that resembled a stock market, where they weren't even often literally trading the bulbs, but just kind of on paper trading the bulbs at higher and higher prices, until, as tend to happen, one day that speculative bubble burst, as people realized, okay, the values we're trading has really, these are just some flowers, and actually, what are these values based on? And people completely lost confidence overnight, and people lost their fortunes overnight. And it's a very famous event in the history of economics, as a really early example of the sort of speculation that you see in stock market bubbles that then lead to crashes. But I'm not going to go too much into the economics. I just wanted to talk a little bit about tulips. One of the things within tulip mania, which was the most valued tulips, were those that had streaks of yellow and white in kind of wild, irregular patterns in the petals, which are known as broken tulips. And the way that this works is that these plants, this was unknown at the time, this was literally just a kind of something that researchers realized in the 20th century, those plants were infected with a virus called, which, when it was discovered, was named tulip breaking virus because of this property. And that where the virus, kind of as it spreads through the flowers and the different cells of the plant, it removes color from the infected cells. And so it creates these wild patterns, this kind of variegation. But it doesn't just it makes the flowers look cool. It does, but it also does weaken the plant. And you probably know that tulips have bulbs where kind of they'll bloom and then they'll basically kind of looks like they shrink back into the bulb for another season. The bulbs weaken over time with the tulip breaking virus, so they don't have an indefinite lifespan anymore. They will die after a few generations. Which is why today there are very few truly virus-broken tulips in cultivation. There are, if you, for example, happen to live in the Seattle area like I do, and you know that just north of us in Skagit Valley, there's a famous tulip festival, and you can see all these different kinds of tulips. You'll see plenty of examples of ones that have streaks of white or yellow in the petals. That sounds like this tulip breaking virus. It's not the same thing. So those ones that you can get commercially, they have in fact been just bred through traditional plant breeding methods, which we'll learn more about in upcoming episodes, in order to achieve that stripy pattern. I did, for a little bit of digging, found out that there does seem to be one truly virus-broken tulip variety that is still commercially available, although it's hard to get your hands on. It's called Tulipa Absalon. That's the name of the variety. And I gotta say, from the pictures, it looks really, really gorgeous, and I would love to get my hands on one. It does, I gotta say, it looks cooler than the streaky tulips that are bred with conventional plant breeding methods. So, in January 2025, researchers Wong, Carrero, and Hillen at the University of Alberta, go back. So I'll just end on this kind of tulip section by saying that in January 2025, so this year, this is, like I mentioned, tulip breaking through viruses was this thing that was first noticed 4 centuries ago. And it's taken till this year for someone to even propose a model of what they think might actually be going on. It's okay, we know there's a virus, but how does it produce these particular patterns? And they basically, these researchers, Wong, Carrero, and Hillen at the University of Alberta, proposed a mathematical model that could account for the pattern of the breaking on the petals, and why the virus would create that specific infection pattern. And so, I'm not going to go into all the details of that, but I'll link the paper in the show notes just to show, just because people can see a phenomenon doesn't mean that we know why it works. Even something I know maybe not everybody in the world knows about tulip breaking virus and the tulip mania, but it's reasonably well known, pretty famous thing within botany, and there's still a ton of open questions. Okay, now dahlias. Dahlias, like tulips, grow from seeds, yes, but also from underground structures. Tulips make bulbs, which are underground storage organs that consists of a shoot apical meristem surrounded by specialist leaves, very much like an onion. So onions are bulbs, tulip bulbs are bulbs, very analogous to each other. If you slice open an onion or a tulip bulb, they all look very similar. Dahlias make tubers, the same sort of structure as potatoes. Tubers are fleshy pieces of root or stem that contain a lot of carbohydrate to store energy and have lots of smaller meristems on them from which new growth can emerge. So the structures are a little bit different, but similar end effect. Underground storage organ that can be dormant and then new growth will emerge when planted, normally in spring. And while tulips are native to Central Asia, dahlias are native to Mexico and Central America. From my understanding, dahlias have long been cultivated in the region, Mexico and Central America, as a food crop. Yes, dahlia tubers are edible, so are tulip bulbs. In fact, eating tulip bulbs that had piled up during the Second World War was part of how Dutch people survived a forced starvation imposed by the Nazis in the winter of 1944. Unpleasant episode. But back to dahlias. Pretty much as soon as Europeans started colonizing the region of Mexico and Central America, they were taken by the beauty of dahlias and built on existing substantial diversity and varieties to create more and more ornamental varieties of dahlia, which has really just continued ever since. Dahlias are maybe not quite in the same league as roses in terms of share of global flower market, but they are definitely, one of the heavy hitters, and they have very passionate fans. Probably unsurprisingly, dahlias are the national flower of Mexico. More surprisingly, given the total lack of historical connection, they are the city flower of Seattle. As far as I can tell, let me know if I'm wrong about this. There was a nationwide craze for dahlias across the US in the early 1900s, including in Seattle, but it's not limited to Seattle. But Seattle was hosting a World's Fair in 1906 and didn't have an official city flower. Not that cities need an official flower, but, didn't have one yet. And some local dahlia lovers thought that an official declaration making dahlias the city flower would be a kind of impressive show of Seattle's cultural sophistication ahead of the World's Fair. Mostly that has not meant very much for Seattle. Unlike, Portland is City of Roses, Portland, Oregon. You'll find tons and tons of roses around the city, kind of rose iconography. You don't see a bunch of dahlia iconography around Seattle. But there is a dahlia show garden in Seattle's Volunteer Park with different dahlia varieties that bloom in August, which is the normal time for dahlias, and it's absolutely gorgeous. So if you happen to be in Seattle in August, go to Volunteer Park, check out the dahlia garden. It's lovely. Something else that I learned when researching this, there is a town in the Netherlands, just to go back to how much Dutch people love flowers, who did not want to miss out on the floral action with dahlias, and since 1936 has hosted a dahlia parade. And this is not just, there's some flowers, and you kind of go through the streets and people hand out flowers or something. No, this is a pretty big deal. It's the town of Zundert. Not sure how to pronounce that correctly, even though I do have Dutch heritage, do not know how to speak Dutch. So it's the town of Zundert, but all the local villages around this town compete by creating massive, elaborate floats decorated entirely with dahlias. And the dahlias, they grow themselves. And apparently, according to the internet, each year, apparently about 6 million dahlias are grown and used in the parade floats. And I really recommend just look up some pictures of this online. The floats are really cool. The parade is called the Bloemencorso Zundert, but I think if you just Google Dutch Dahlia Parade, you'll get it. So I'd recommend that. These days, there are probably thousands of distinct varieties of dahlias, and by varieties, I mean distinct genetic lineages. And the aesthetic diversity of dahlias is much greater, I would say, than for most ornamentals. There are different varieties of dahlias that just, at first glance, don't look like they're from the same species at all. And there's certainly this diversity, a lot of it has come from small-time breeders working in the same sort of way as people have done for centuries. So we'll learn in upcoming episodes about some of the more modern plant breeding approaches and methods that have been developed in the 20th century and 21st century. Particularly, obviously, we'll get to GMOs, but we'll learn about some other stuff along the way. But there's also what people have done for a long time, which is plant a bunch of different seeds, make kind of crosses by transferring pollen between plants, and kind of just see what you end up with. And one of the great things about dahlias is because they produce these tubers, the tubers basically allow you to, there's no kind of production of seed and transfer of genetic material between different parents that goes into producing a seed. It's just a clone. The tuber, like with potatoes, the tuber is just a piece of the adult plant. You can, in fact, take the tubers, break them apart into smaller pieces, and propagate basically clones of that same adult plant. So once you get a nice dahlia variety that has beautiful flowers that you love, you don't risk kind of losing it again as it interbreeds with other individuals. You can just propagate that dahlia through tubers indefinitely. That also make dahlias excellent candidates for mutation breeding, which we'll learn about in 2 episodes' time. But it's basically deliberately exposing plants to mutagenic radiation or chemicals that are mutagenic, which modifies DNA sequences randomly. And you can do this, you're going to take a Dahlia tuber, like break it into hundreds of small pieces, expose it to this mutagenic radiation or chemicals, and then kind of let those tubers, like plant them, see what the flowers look like, and there's a good chance you'll get at least one that has some new, interesting flower phenotype, and then you can just start propagating that, and hey presto, you've got your new Dahlia variety. And so, that's mutation breeding has been particularly successful in the Dahlia breeding industry. So let's connect this back to the original question. Like tulips, there are varieties of Dahlia with variegation, meaning stripes and dots in the flowers that lack pigment or have a slightly different pigment. And unfortunately, sort of short answer to this is it seems, as far as I can tell, there is no viral infection that causes this. It's just one of the many, many different varieties that popped up as people have been breeding Dahlias. They are just genetic variations. So, sorry, I could have answered this question with just a simple no, but where would be the fun in that? And I'll link some of the references that I was drawing from in the show notes. Okay, let's do our final question. Can we GM a mango so that it has edible skin like an apple? So I want to thank Mechanical Bees on Reddit for this question. Can we GM a mango so that it has edible skin like an apple? This time I'll start with the short answer, probably yes. So let me share some of my thinking about this. Firstly, what is it that makes the mango skin kind of unpalatable? There are a few different pieces to this. One, it's quite thick, like quite physically difficult to eat. So that would be one issue. Then it's just quite bitter. And then kind of sub bullet point of bitter, there's a specific bitter compound in this peel called urushiol, which is particularly problematic. It is named for the Japanese name for the lacca tree, and lacca trees and mango trees are in the same family, that the Anacardiaceae. Most of the plants in this family produce urushiol in some of their organs. The family includes cultivated plants like mango, cashew, and the lacca tree, which is used to make true lacquer used in Japanese lacquerware. The family also includes poison ivy, poison oak, and poison sumac. And so that might give you a hint about the problem here. Urushiol irritates skin by triggering an inflammatory immune response, or what we call an allergic response. In those that have sensitivity, this is potentially life-threatening. For most of us who are not kind of hypersensitive to it, it's just annoying and a bit painful. So that hypersensitivity, when we say allergic, we actually mean a hypersensitivity, but an allergic reaction just means it triggers an irritation that is based on basically a gentle immune response. That's an allergy, is kind of a inappropriate immune inflammation. Pretty much everyone gets it to some extent, both for poison ivy and for mangoes. But the amount in mangoes is kind of less problematic, and there are kind of different versions of urushiol. And the one that is in mangoes is one that tends to be less irritating, less triggering of allergic reactions than the one that is in poison oak. So that's why mangoes tend to be less problematic for people, but the chemical is particularly concentrated in the peel. So for most people, the amount that you get exposed to just from eating the fruit of the mango isn't enough to cause much of a reaction, maybe like a slight tingling. People who have what we call, like in everyday speech, an allergy to the mango, there's a whole range of how sensitive they are. But for most people, if you ate a whole mango peel, you would not enjoy it. You would have some sort of unpleasant, irritating reaction. So if you were to make a mango with a peel like an apple that you'd want to eat, you would need to do quite a lot. You'd want thinner skin that is less bitter, but you'd definitely want to get rid of the urushiol. So I found a paper in 2020 that they published the mango genome. It was led by a large research team in Hainan, China. And as part of the work, they looked at transcription, so that's gene expression in different tissues, and they pinpointed a set of genes that encode enzymes called chalcone synthases. And that particularly they found the chalcone synthases that are expressed more in peels. And just based on what we know about the functions of similar enzymes known from other plants, they're pretty sure that these are the enzymes that are responsible for making the urushiol and the peel. So if I wanted to make a transgenic mango that had peel that was more palatable and less irritating, what I'd want to do is knock out or break those specific chalcone synthase genes in the mango. And now that we know what those genes are, it's a lot easier to do because, we'll dive into all of this when we explore how you make GMOs. But obviously, you kind of had to know what you want to add if you're trying to add in new genes, or what it is you want to break or modify in the existing genome. Like, if you don't know that, you don't have any way to begin your project. So knowing that is not everything you need, but it's a very important first step. Bigger problem that I could see is that no one seems to have made a transgenic mango, meaning an adult mango tree that has had its genes modified using laboratory methods. Like many trees, mango is hard to do breeding with, and most mango varieties around the world are random mutations that appeared, and then cuttings from the tree have been cloned over and over. Kind of just like you can take tubers and break them apart and grow new dahlias from them. With a lot of fruit trees, you can just take various pieces of the tree, and with a little bit of gentle coercion and sometimes a little bit of chemical help, you can get those little pieces of tree, those cuttings, to grow into full adult trees. And that is how most fruit trees are propagated, not from seeds, but from cuttings of adult plants. And that's great in some ways, but it also means it's hard to do breeding with those trees. It's hard to kind of create new varieties. We will see that making a transgenic plant takes a lot and mostly involves grinding the plant into cells and using one of a few methods to get DNA into those cells and then getting the cells to turn into adult plants. And those steps can be very easy for some plants and seemingly impossible for others. And I suspect for mangoes, it's both difficult but also just not something that anyone is pouring tons of money and attention into is the bigger problem. So like a method of how exactly to do this for mangoes is lacking. Mango is a big crop. It is pretty commercially successful all around the world, but it doesn't come close to the global market value of staples like rice and potatoes. So there's just not as much economic incentive to bother trying to work out how to create transgenic mangoes. So that does not mean that it is impossible. But let's just say maybe it is impossible and we can't, for some reason, just the normal methods that have worked for many other plants just won't work for mangoes. We cannot genetically modify them. Then you'd have to rely on conventional breeding. But mangoes, like, as I mentioned, kind of basically all fruit trees, are propagated by cuttings, not seeds. Because, and just to dive into this a little bit more, the seed will not give you a tree that looks like the mother tree that produced the fruit it came from. The seed has a combination of maternal and paternal genetic material. The fruit around the seed just comes from the mother. And so and generally the fruits are what we're interested in, but all the other organs of the tree are what we call the mother, and then the father tree just gives its pollen. And just to I know I'm kind of complicating things in case you're wondering, there's mother and father trees. Most plants are hermaphroditic. They produce male and female organs. Some aren't. Sometimes, particularly with fruit trees, you do sometimes get male ones and female ones, and you'll see only the female ones produce fruits. The male ones just produce pollen. Anywho, there's a bit of but in general, the seeds are the end result of that sexual reproduction. The seeds will have a combination of genetic variation from both parents, and so will not look like the mother tree. And so that can be, if you have a variety of mango that is great, and you love the fruits that it produces, you want to just reproduce that plant via cuttings to always make sure you get identical fruits every time. And we'll learn more about that in the coming episodes if you're like, wait, that was too much and I'm confused. And so if you're wondering, like, is this just an issue for fruit trees? No, it's an issue for all plants. But in most crops where breeding based on sexual reproduction is done, this problem has been evaded by deliberately inbreeding plants for many, many generations to the point where there is no genetic variation, or whatever genetic variation is there is the genetic variation you kind of want and are controlling. But that takes many generations of inbreeding, which is fine if you have like wheat, which you can actually get 2 generations out of it in a single calendar year with the right coaxing. You can breed, you can inbreed that wheat 10 generations in 5 years. But a mango needs many years to get from seed to sexual maturity. So it's just like slow and painful to use that kind of method. And that's why just in general, this is true for all fruit trees. Trying to do conventional breeding with them is slow and painful. To give an example, there's this new apple variety, the Cosmic Crisp, that was released in Washington State in 2022. I think it is more widely available these days. I'm a big fan of it. It took a huge team of agricultural scientists 22 years to make this one apple variety that is essentially just a cross between 2 prior apple varieties. So it's far harder to breed new tree fruit varieties using conventional breeding methods. And the same, I'm sure, is just as true of mangoes. People are breeding mangoes. There are teams of people around the world doing this. It's just like slow and painful, and like I said, there's not as much economic kind of interest around mangoes, so there's not as much effort going into it. And there are, also to point out, there are already many different varieties of mangoes that exist. It's not a kind of urgent pressing problem. But as we'll see kind of in the coming episodes, there's all sorts of reasons why, even when you have like a perfectly good variety of a plant, you always kind of want to create more and have more options on the table, particularly in case there are diseases that come up that threaten the existing varieties. Okay, anyway, so plant breeding could be an option. It's going to be slow and painful, but it could happen if people really were determined to get a mango without urushiol in its peel. And there's a precedent for this that I thought was a fun opportunity to bring it up. That was a pretty... actually, there's like quite a lot of interest in breeding out unpleasant chemicals from plants in the 20th century, and one of the most successful examples of this is canola, which canola is Brassica rapa. It's from the mustard family, and it is like has long been known for its oil that comes from its seeds, but that oil was more just used as an industrial lubricant or for other kinds of purposes, not for eating, because it's very, very bitter, naturally, because of a chemical called arucic acid. And some researchers in the mid-century in Canada were like, we are not going to settle for that. We think there's a lot of this plant grows really well in our climates and our soils, and if we can just make this oil edible, it could be an amazing oil plant for use to make cooking oil or eating oil. And what they did was use conventional breeding methods to completely eliminate the arucic acid, or to get it to such low levels that it's not bitter anymore and it is perfectly edible and pleasant. And the product of that breeding was the kind of plant and oil that we now call canola. And this is like fun bar trivia fact that canola is an acronym that stands for Canada Oil Low Acid. So the plant is Brassica rapa, but the particular variety that can be used in cooking is called canola. I think both because they wanted to have like a nice marketing gimmick, like indicate that this is a new product. This isn't the same old Brassica rapa that you know, but also because the conventional name for Brassica rapa in English, at least as a crop, was rapeseed. And that is actually still how it's mostly known in the UK and in some other areas. But like rapeseed, you can imagine, is a name that has unwanted connotations. And so canola just sounds great. And so canola is what's used more in North America. Anyway, so that's like, there's precedent there for the sort of, you could imagine following a similar approach to get rid of the urushiol in the mango peels. But that's not been done yet. So in the meantime, what do you do with all the discarded mango peels? researching this, I did find some papers by researchers such as D.V. Sudhakar at Indian Agricultural Research Institute in New Delhi, developing methods to take discarded mango peels from mango processing and extract usable chemicals from them to reduce food waste. And just as a reminder, that most crops don't make it to the end consumer in an unprocessed form. So there's a lot of waste that builds up in the processing of plant products, and it can be like great to see, like, what can we do with that? How can we make sure that doesn't just go to waste and pile up and decompose and release methane gas, which is a powerful greenhouse gas. So just a shout out for all of those diligent workers working on these not very glamorous questions like how can we process and get valuable products out of mango peels. And yeah, just to kind of remind you that the original question was about, like, could we GM a mango to have an edible peel? I think yes. You'd have to get that genetic modification method to work, which would probably just mean like testing out a bunch of parameters until you get one that works. And then we kind of know which target genes we'd want to go after. I think it would be doable. I just doubt that anyone would be that motivated to do it. But we'll see. It'd be very fun to see that in a few years, edible kind of mangoes you can eat like an apple coming onto the market. Okay, so I think I'll wrap up here because I think I've been talking for a pretty long time. If you hated this, don't worry. We'll be back to a regular scripted narrative episode after a 2-week break so I can catch up with writing and recording. If you liked it, let me know. I could definitely do more Q&A episodes based on your interest, dear listener. To let me know by emailing orlando@modifiedpod.com, or that's orlando@modifiedpod.com, or you can find my socials at modifiedpod.com. That's the website. That's also where you can find the show notes. And just as a reminder, I'm Orlando Delange. This is Modified. As always, please do subscribe, leave a review, and tell a friend if you're enjoying the podcast. And I hope you'll tune in again soon. Bye.